I'm Bruce McEwen, a professor here at Rockefeller University, and I want to welcome you to the 48th Holiday Lecture. I'm also very, very pleased to be introducing Paul Nurse today for today's lecture. It's not only because Paul, an Englishman, is a great scientist, a great communicator, a Nobel laureate, and our president. It's also because Paul Nurse's personal history parallels in some ways the history of the English scientist who presented the first holiday lecture, then called the Christmas Lecture, in London in 1860. This was Sir Michael Faraday, and the lecture was called Chemical History of the Candle. Faraday, the son of a blacksmith, grew up near London, working in a number of ordinary jobs until he became an assistant to Sir Humphrey Davy at the Royal Institution in London. Working hard and showing great promise, Faraday worked his way up and became an independent scientist and one of the great chemists and physicists of his time. But Faraday never forgot how he got where he was and created the Christmas lecture tradition in London to reach out to young people. The late Professor Alfred Mirsky, a New Yorker who trained at Cambridge and then was a professor at Rockefeller for many years, was a great admirer of the Christmas lecture tradition in London, and the university, Rockefeller University, created the Holiday Lecture Series in 1959 as a means for reaching out to potential young scientists. As I said, Paul Nurse's lecture this year is the 48th in the series. In 1992, the Science Outreach Program was established under the able directorship of Dr. Bonnie Kaiser and then President Torsten Wiesel. And the holiday lectures found their place as the capstone event in the Science Outreach Program. Our Science Outreach Program offers the exciting environment of Rockefeller to both selected teachers in a 2-year partnership with laboratory and to motivated and talented high school students. About 700 high school students and 100 teachers are outreach alumni. About 1 in 5 students gained at least semifinal status in science fairs at the level of the Siemens and Intel competition. In fact, just this past month, our science outreach student, Alicia Darnell, sitting in the front row, garnered 2nd place in the Siemens competition. You want to stand up? We are very proud of all of our student alumni, some of whom have even returned to Rockefeller for their Ph.D., 1 person, M.D., Ph.D., 4, and postdoctoral work, 1 so far. Teacher alumni are equally impressive. 12 teachers have been promoted to principal, assistant principal, or science chair. At least 6 have won awards for excellence in teaching. Many teachers have developed and instituted new advanced placement and or research courses in their schools, and 1 just sold his website, My Biology, to a book publisher. A number of teachers have pursued or are pursuing advanced degrees in education or science education. Now, as to our speaker today, as I said, Paul Nurse has a story that in some ways parallels that of Michael Faraday. He grew up near London in a working-class family, and with his own talent and motivation, aided by the mentorship of a number of exceptional teachers, he developed his talents in biology. As Faraday had done, he took advantage of the opportunities that opened up to him as his talents became evident. At key stages in their respective careers, both Paul Nurse and Faraday profited from being in stimulating and exciting scientific environments where the sky is the limit. Rockefeller University is such a place. I know this personally because I had that influence on me when I came as a Midwesterner from a small college to study here in 1959, where I was fortunate to have Alfred Mirsky as one of my mentors. So, as I said, it's a great, great pleasure to introduce Paul, and the title of his lectures is 5 Great Ideas of Biology.
Well, Good morning, everybody. It's a pleasure to see so many of you here so soon after Christmas and on a rather grey day. It's nice to see you all. We started with Benjamin Zephaniah. That was the poet you saw gyrating on YouTube a few minutes ago. He had some lyrics about genetics, which is why we put him up there. And genes and genetics is going to be one of the ideas I will talk about today. But there's going to be 4 others, a total of 5. And 4 of those ideas will be rather familiar to you. But I'm going to ask you not to get bored with them right at the very beginning, because I'm going to try and present them in perhaps a different way from how you've heard about them in the past. And those 4 ideas, I'm going to focus on history, where the ideas came from. You're going to see a number of photographs of rather serious-looking 18th century and 19th century gentlemen at different stages during this talk. I'm going to talk a bit about the philosophy behind those ideas, and I'm going to talk a little bit about the relevance of those ideas for today and for ourselves and for the issues of today. And I think what you will see, or I hope you will see from this, is that science is truly revolutionary. We often think of politics as being revolutionary, but in fact science is a truly long-term revolutionary subject, one that changes constantly the way we view the world, the way we view ourselves. 4 of the ideas I said are familiar. The 5th idea will be less familiar, and I'm going to leave that to the end. So my talk will be in 2 parts, and 3 ideas in part 1, and 2 ideas, including a new developing one, in part 2. Now, you know, biologists tend not to talk about great ideas. In this sense, we are different from the physicists. The physicists like to talk about great ideas of their subject, you know, like Newton's laws of motion, thermodynamics, conservation of energy, Einstein's views about relativity and the like. Biologists tend to like details. They like describing things. They like noting the details. And as a consequence of that, sometimes the ideas get lost. You know what us biologists do? If we were in the 19th century, we'd have collected insects and counted how many hairs there were on a beetle's leg, for example. If we were an ecologist, we'd list the numbers of species you'd find in different environments. If you're a geneticist like me, we sequence genes. Lots and lots of details and information. Those details are very important. But sometimes the ideas behind those details get lost. So I've put together 2 talks today, which will focus on those ideas, those main ideas of biology. They're my choice. There's others which are equally important, but these are the ones I'd like to introduce to you today. So the first idea is the cell. The cell as the basic unit of life. And we can start with a definition. All life is composed of cells, and the cell is the simplest unit exhibiting the characteristics of life. 2 ideas there. The cell is the basic unit, structural unit of life. Secondly, it's the basic functional unit of life. One way to view it is as life's atom. Just like the atom is the basic unit of matter, the cell is the basic unit of life. Now, this idea took many years to develop. It started in the 17th century, and it took nearly 200 years for it to be fully appreciated. And as is often the case in science, it was technology which begat discovery. What do I mean by that? What I mean is that it was using technology which allowed scientists to have a different view of the world. Just like radio telescopes allowed the spectrum of radio frequencies to be investigated, in the 17th century, it was the invention of the microscope. And you'll see on the left there, on this slide here, a picture of a 17th century telescope, a 17th century microscope, invented together with the telescope in Holland in the beginning of that century. But the person who used it first in any extensive way to look at biological things was Robert Hooke. And Robert Hooke was an interesting character. He was the experimentalist for a society called the Royal Society. And the Royal Society is a scientific academy, a scientific society based in London, but was set up in 1660. But the point about it is that it was mainly composed of gentlemen. And of course, gentlemen did no work. And so they hired somebody to do the work for them. And the person they hired was Robert Hooke. He was the experimentalist for the Royal Society, and he did experiments that the gentlemen could then talk about. And he was a polymath. He looked at all sorts of different phenomena, from physics through chemistry through to biology. He had a huge argument and row with Newton, with Isaac Newton, and as a consequence of that, his reputation was eclipsed somewhat for a couple of centuries. But he was an interesting person, and he turned the microscope to plant material, and he took a cork, a piece of cork, and I don't know if it was from his wine bottle or not, but he took a piece of cork, and he took a little slice of it, and he put it under the microscope, and that, you see here what he could see. And this is what we would see today if we used a modern microscope. So it's pretty similar. And this is a drawing of a book he published in 1665. And what struck him was that there were a lot of boxes he could see in the slice of a cork, and it reminded him of a sort of map of a monastery full of monks' cells. And as a consequence, he called them cells, and this is the origin of where cells came from. Now, this depended, as I said, upon the invention of the microscope. And just like technology often begats discovery, a second thing that often happens in science is that once one person discovers something, everybody discovers it. But it needs somebody to discover it. And within 10 to 15 years of these illustrations like the one on the left were being seen. This is a picture from a book published in 1682 from a guy called Nehemiah Grew, who was a doctor, also in England. And this is a wonderful picture, don't you think, of a stem section of a vine. This is what it would look like today. I happen to have got this book, which this was published in, and it's this book here. And if anybody's interested, I can show it to you afterwards. It's full of pictures like this, and I think they're absolutely beautiful. Others also began to look at biological material. This is Antony von Leeuwenhoek. Antony von Leeuwenhoek was a bit like Robert Hooke. He was not a gentleman either. He was a draper who worked in Holland, in Delft, and he sent all his observations again to the Royal Society. And what I'm showing you here is the first picture of single-celled life, because he discovered that you can get living things that are just made up of a single cell. And what he did was he took a scraping of his teeth. Any of you could do it today. And he put the scraping on a microscope slide. He looked under his microscope, which actually was a very powerful but single-lensed one, and he saw all these little swimming animals, he called them animalcules, swimming about, all from his teeth. You've got them swimming about on your teeth now, by the way. And it's charming. Look at Fig B over there. You see he sort of drew what they did. There's one doing a loop-the-loop, this bacteria underneath the microscope here. And this is the first demonstration of single-celled life by Antony Leeuwenhoek. Now, we don't really know what Leeuwenhoek looked like, but Leeuwenhoek knew the painter Vermeer. Some of you may know about Vermeer. He was a Dutch painter also of the 17th century, and he died in his 40s, and he left a will, and the executor of the will was Leeuwenhoek. And I like to imagine, and I have not one piece of evidence in favor of what I'm about to say. By the way, scientists sometimes do that as well. They say things without any evidence whatsoever. We shouldn't be doing that, but we sometimes do. And I like to think, because Vermeer very unusually did two paintings of a scientist The same scientist, an astronomer and a geographer, and this one here is in the Louvre. I like to think that this might be Leeuwenhoek, who was his friend living in Delft. Well, that was the discoveries of cells, but in the 1600s. And it took a little while longer to see cells in animals, because they're not so easy to see. Malpighi and Leeuwenhoek himself saw blood cells, but it took really about 100 years improvement in microscopes and sectioning tissues and the like to be able to see that actually cells were everywhere. Cells made up plants, cells made up animals, and we see these single-celled microorganisms that I've just shown you on a previous slide. And so around the beginning of the 19th century, the idea of the cell doctrine was born. Interestingly, not many years after Dalton and others had postulated the existence of the atom. I remind you, I was saying you could see life as the cell as life's atom. Just at this time, physicists and chemists were thinking about what was the basic unit of matter, and maybe that influenced how biologists thought as well. Anyway, Theodor Schwamm, the zoologist, and Schleiden, the botanist, proposed that all organisms are composed of essentially like parts, namely of cells. And that's up here. So this was really the birth of the cell theory. A major milestone in biology, but not one that's generally recognized. Then it took another German, this is Rudolf Virchow, a few years later. He invented pathology, by the way, in the mid-19th century. He emphasized that cells also are the basic functional unit of life. What do I mean by that? Well, it's written here. Every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life. Complicated sentence, but very important sentence. What it says there is that the cell is the basic unit of life because it has the properties of life. And this is a fact statement that I'm going to return to several times during this talk. And so here we have the cell theory. Cell as a structural unit, cell as a functional unit. Now this really does underpin much of biology. Why do I say that? Well, here is the cell, and we know much about it, and I wanted to point out here that Rockefeller University played an important role in taking cell biology into the modern age in the second part of the 20th century. And here we see a modern picture of a cell with a nucleus here, the DNA, I'll talk about that later, mitochondria here, the powerhouse of a cell, endoplasmic reticulum in blue all the way around here. And these four scientists, from Palade to Blobel, all played very, very key roles at Rockefeller University, in this very place where you're seated, in developing the cell as a fundamental idea in biology. Now, cells are the basis of growth and development. And I'm going to show you a little movie here, taken here at the university, showing cells dividing. Because a cell will undergo cleavage from 1 to 2 to 4 to 8 to 16. And this is the basis of all reproduction, including reproduction of our cells, and all growth. So let me just start this movie. Look at, I'm going to play it twice probably, this cell or this cell, and see what happens. Now, look at this guy here. You see, bang. These are the chromosomes, and then they separate into two. Same one, that's happening here. Dividing into two. Let's just go back and look at that again. Focus on that cell and the cell down here. And you see the chromosomes appearing and then dividing into two. But the emphasis I'm trying to give you is that this is the process of division of a cell from 1 to 2. Incidentally, this is what I myself work on. This is my obsession of my life, you know. My family, two of whom are sitting over there, actually think I'm a rather sad individual, having committed about a third of the century of my life. They're over there, okay? They think I'm a rather sad individual, having sort of devoted so much of my life to this process. But I want to point out to them that they are formed as a basis of this, you know, cell division, okay? Now, that cell division gives rise to an embryo. This is a mammalian embryo here, and you can see here, if you look carefully, these little black blobs. These are actually individual cells. But I've got here a movie of a frog embryo. This is a single egg, just to show you how a little embryo, a tadpole, can develop from cell division. So it's undergoing some movement. Now look at that. That's the first division. Do you see that? Now it's undergoing, see there's 3 or 4 cells here, all cell division. Many, many divisions. You see them? Yeah? Great. Now, see them all here. By the way, note this. I'm going to say something about stem cells in a minute or two. But see all these lots of little cells? Then they get so small you can't see them anymore. And then you get lots of movements of this embryo. It's very active in there. This is what, if you speed up, what happens with a frog development. Can you see? Yeah. It likes to spin, you know. Likes to spin. And at the end of that, look at that. You know, you've got something that looks recognized. You see there's a nerve cord going down the bottom here, you know, this frog. There's a bit of an eye developing here. It starts to twitch again. See the tail, you know. Then it doesn't like it in there very much in the egg. You see, it sort of twitches around a bit. Twitch, twitch. And it's about to break free. Look, very busy, bang. Right. So this all happened as a consequence of division of that single cell. And one thing I want to sort of emphasize for you is that that single cell had the ability to change, what we call to differentiate into all the different cell types that make up that embryo and then our bodies. So it can make bone cells and nerve cells and muscle cells. All of these different sorts of cells can be made from this single cell. And if I haven't convinced you that cells are interesting, then I'm just going to put this slide up here and remind you, every one of you in this room was once like this, a single cell. This is a mammalian egg being fertilized by sperm. Every one of you was once like this. Are you not interested? I hope you're interested. So that's my first idea, the cell. The basic unit of life. It underpins all reproduction and also development. And it's still relevant today. And many of you will have read about stem cells, I'm sure, in the newspapers. And I mentioned stem cells in passing when I was talking about the embryo. What are stem cells? Stem cells are cells which have the ability to change or differentiate into different cell types and different cell tissues. And in particular, embryonic stem cells, like those ones that I showed you in the frog, can differentiate into all cell types. They are what's called totipotent. That means that they're potent, they can change into all cell types. That's where the word toti comes from. And these are very interesting for 2 reasons. One is it's biologically interesting. How does this change occur? Because it's the basis of how we develop. And secondly, it's likely to be medically relevant, because there's a whole class of medical problems, medical diseases, which are a consequence of degeneration, like muscular degeneration, like nervous tissue degeneration. And there's very little we can do for patients like that. But maybe, if we can make use of stem cells, we may be able to differentiate these embryonic stem cells into cells which will replenish damaged nervous tissue and damaged muscle tissue and the like. And it may give some hope to patients of this sort. And as you know, this is a contentious area because to derive these embryonic stem cells, we have to take a very, very early human embryo, looking like a collection of these cells that I showed you with the frog, and we use those for deriving the embryonic stem cells. So this is an issue that has been talked a lot about, especially in the United States. And indeed, at the moment, the government does not allow federal funding in some areas of research. Private funding is allowed, and we indeed do this work on this campus here, but not federal funding. But it does have the potential, but at the moment we have no more than potential, but it does have the potential to providing some potential cures for some of these diseases. So that's the cell, still relevant today. The second idea you're probably more familiar with, and that's the gene, which forms the basis of heredity. Now, From ancient times, it's been observed that particular characteristics of an individual appear to be passed down from generation to generation. So if you have tall parents, you tend to be tall. If they have blonde hair, you tend to have blonde hair, and the like. And so this has always been fascinating. How is it that offspring resemble their parents? Well, not much progress was made, even though this was thought about for actually many centuries, until, again, the 19th century. And during the 19th century, there was a lot of research going on in plant breeding, largely to see whether you could improve plants for crops and the like, but it is also basically important for basic science to try and understand the nature of heredity. But the truth was, not very much progress was being made, because what plant breeders found was that it was all very complicated. They would take two plants which sort of were the same species, but they would differ, say, in the the colour of their flowers or the height of the stems or something of that sort. And then when they crossed them together, they would find that certain characteristics would just disappear in the first generation, and then in the second generation they'd reappear again. And there seemed no rhyme or reason to what was actually happening. And so this was a very, very confusing area. And it needed really a genius to sort it out. And the genius was a guy called Gregor Mendel. I know him as the gardening monk, because he was a monk. In fact, he became an abbot, but he was also very interested in plant breeding and in gardening. Now, Mendel, he himself also came from a poor family, like Hooke and Leeuwenhoek. And he was trained as a physicist to become a physics teacher. And he entered a monastery. And in the 19th century, monasteries, certain orders of monks had quite a lot of interest in science. And he joined a sort of scientific sort of monastery in a place called Brno in the Czech Republic. And he was there to teach the local school children physics. And indeed, he was originally a meteorologist and an astronomer. I've seen his telescope, for example. It's on tour of the United States at the moment. I saw it in Chicago in the Field Museum. But he also monitored air pressure as a storm went through. And was one of the first to show that air pressure dramatically went down when a storm goes through. But despite being trained as a physicist, he turned to gardening and tried to work out what the genetics was behind plants. But because he was trained as a physicist, I think he approached it in a different sort of way from most biologists. And one thing he did was to actually count what was going on. So when he made crosses between plants, he would count the numbers of offspring and the types of offspring that he saw, which curiously to us today, other scientists hadn't done. They just sort of described it qualitatively. They'd not described it quantitatively. And I'm sure that was influenced by the fact that he was a physicist. So I think there were three reasons for his success. One is that he chose material that he could understand. Now, why do I emphasize that? Well, he did trial experiments with about half a dozen different plants and found most of them he couldn't really understand what was going on. Now, you may think scientists are objective and just observe all facts and then just sort of try and make sense of it. Think again. What scientists do is actually try and study things that they can understand. It's the art of the soluble, as Peter Medawar, a scientist philosopher, said some decades ago. And what Mendel did was to reject most of the plant species he was trying to study because they were too complicated. And he focused simply on this plant here, the pea, sweet pea, because he could understand it. And he grew, this is a picture from the Mendel's monastery in Brno, and he had this big garden. And you'll see where this garden here, this part of it, was all a gigantic greenhouse, which is where he grew all his plants. And in it, he had these sweet peas. And the reason he chose sweet peas is because he could make some sense of it. And I want to emphasize that, because biology is complicated. And sometimes you need certain model systems which sort of expose the ideas in a simpler way. And then when you've made progress with those simpler systems, you can move out into the more complicated ones. The basic issue here, since many of you will be familiar with genes, is that in the sweet pea, he chose characteristics determined by single genes, whereas in the other plants, there were many genes involved, and he just couldn't sort it out. So that was his first thing. He chose material he could study. The second thing he did was to record accurately what he saw and counted them. I've already mentioned that, and I think the physics background influenced him there. And the third one, which was also a bit unusual for biologists, is he generated a theory or a law to explain his observations. And again, that's the sort of thing that physicists tend to do. So what he observed is that in the first generation after a cross, if he crossed different sorts of plants, you can see the different plants here. We've got tall ones, for example. He crossed tall ones versus dwarf ones, ones of different flowers, ones of different seed colours of flowers, ones of, with different seed colours. I'll show you one of those in the next slide. When he crossed these, he observed frequently that in the first generation of seeds that were produced, one of those characteristics would just disappear. So he introduced the idea that, there may be one character that was dominant over another. And then the second thing he observed was that when he crossed those offspring with each other, the original variety, the two, say the tall plant and the small plant, re-emerged. And this is a classic case. I hope you can see the colour here. What he did is, and he described his paper in 1865, he took a yellow-seeded plant and a green-seeded one, crossed them together, and everything was yellow. When he crossed all these together, he got a green one out, though mostly they were yellow, and this kept happening. And so he noted not only that this happened, but that the green one was at a very, very clear ratio. In fact, only 1 out of 4 were green, but it was very, very accurate, 1 out of 4. And he began to notice, and I don't want to teach you all genetics, so I haven't time to do that, but he noticed these ratios, and that's the only thing I really want you to remember. And he saw very precise ratios. Here's 1 to 3. In different sorts of crosses, he saw 1 to 1. In other cases, he saw 1 to 2 to 1. Very simple ratios. And he got very good numbers to support it. It was very, very precise. In fact, so precise that a statistician in the middle of the 20th century called Fisher reanalyzed the data and concluded that Mendel's data was too good to be true. And he concluded that somebody must have fiddled the results. But of course, he didn't blame Mendel, he blamed the gardener, saying that the gardener knew what Mendel wanted to see, and so when he counted the seeds, he saw the right numbers. I have no idea if that's true either. But what is true is that the precision was really rather good. So as a consequence of this, he concluded that it looked to him as if the basis for heredity were particles that were being handed down from generation to generation, particles that unchanged, and he made a proposal that in any particular plant, for a particular character, like the colour of the seed, there were 2 particles that were important. And it was the interaction between those 2 particles that determined whether you had a green or a yellow seed. And if, for example, you had a particle that was green and a particle that was yellow, then the yellow one was dominant over the green one. But when you now segregated the particles in the next generation, that single green one had 2 green particles, whereas the others had at least 1 yellow one. I think you can sense, without me actually going through it, how that might give rise to these sort of very constant ratios. And another notion in his ideas was that these particles were, in some sense, carrying information from generation to generation, which determined the characteristics of those plants. Now, this was a brilliant piece of abstract reasoning. It really was. It was brilliant. It was totally right, published in 1865, but it had one consequence. Nobody was interested. It was completely ignored. He sent it round to famous scientists of the day. People think it may have even gone to Charles Darwin. I'll be talking about him in a moment. And they all ignored it. They took no notice of it. So he gave up science a few years later and became an administrator. You know, that happens too, I have to say. and scientists become administrators. He eventually became the abbot of the monastery and argued with the government about taxation for the last decade of his life. And then his idea was rediscovered. It was rediscovered by 3 other groups simultaneously who got similar results around 1900. They all started to write their papers, and then one of them noticed that Mendel had written it all before 35 years ago. Can you imagine how galling it must have been for them? Here they were thinking they'd made a great discovery, and here was this gardening monk in Czechoslovakia, or the Czech Republic now, who had made all the discoveries, got all the maths right, and sorted it all out. So they all had to reference the gardening monk Mendel when they wrote their 3 papers. Now, why was it that there was a change then, that it became acceptable? It became then, everybody believed it in 1900, 1901. Nobody believed it in 1865. Well, nobody really knows, but again, I'm going to give my view of it. Because between 1865 and 1901, chromosomes were discovered. And chromosomes, this is actually a picture from the time. This is an onion root tip. And you can see the chromosomes here. And every time a cell divides, and do you remember I showed you that in that movie a little while ago, these little strings appeared inside the dividing cell. And people began to think that maybe the hereditary determinants were located on these strings or on these sort of chromosomes. And then Van Beneden, a scientist called Van Beneden, studying a nematode worm, once again, the choice of the biological material really matters. And this nematode worm only had 4 chromosomes, so they were easy to count. You know, if we go back to here, it's not so easy to count how many chromosomes are on in this onion tip. You go over here, there's 4 chromosomes, easy to count. And what he noted, Van Beneden, was that when the nematode worm made gametes, eggs and sperm, instead of seeing 4 chromosomes, he saw 2 chromosomes. Now, I want you to take you back to Mendel, because what was Mendel saying? What Mendel said was that each determinant was like the height of a flower or the height of a plant or the color of a flower was determined by 2 particles. And that when a plant produced its gametes, only one of those 2 particles would go into the gamete, okay? And then when that gamete fused with another gamete, you would return the number from 1 back to 2. These chromosomes were behaving in exactly the way that Mendel's determinants were thought to behave. Because these chromosomes, there were 4 in the adult, and each gamete only had 2. So suddenly, the abstract reasoning, if you like, of Mendel was transferred into translated into concrete reality, into concrete observation with what was happening with the chromosomes in the nematode. And that's another interesting psychological factor about science. Abstract reasoning only takes you sort of psychologically so far. You need observation and experiment to actually really feel and concrete explanation. And I think that Mendel's abstract reasoning was just one step too far. And when you had the chromosomes, you felt much better about it. So what are these chromosomes made of? Well, once again, I can turn to Rockefeller, because this is a modern picture of a chromosome, and each of these sort of strings is made up, of course, of DNA, deoxyribonucleic acid. And DNA as the hereditary material was discovered in Rockefeller University. And that may really surprise you, because mostly when people think of DNA, they think of Watson and Crick. And Watson and Crick discovered the structure of DNA. I'm going to say something about that in a moment or two. But the fact that DNA was the hereditary material was actually discovered here in 1944. Again, in exactly the place where you're seated today, by Avery, MacLeod and McCarty. And I want to say that Mac McCarty, was still working at this university only 1 to 2 years ago. When I first came here, I was introduced to this charming old man, and he said, "My name is Mac McCarty." Now, I learnt about what McCarty did in late 1960s when I was a student, and I have to say, I thought he was already dead then. And here I was, rediscovering Mac McCarty in 2004, And I was just astonished. And all I did is I remember my mouth opening and saying, "Are you the Mac McCarthy?" And of course, he was the Mac McCarthy. Great scientist, still working with great enthusiasm in his 90s. So DNA was discovered in Rockefeller University. The structure of DNA was discovered in Cambridge and in London, but it's a structure in 1953 that we tend to think of rather than the discovery of DNA itself. And it was made using the pneumococcus microorganism. I don't have time to go through that, except to tell you that what they did was to transform the characteristic of one microbe into another using pure preparations of DNA. And what we now know is, and you can see it here in this sort of textbook illustration, that a cell has got a nucleus. Within the nucleus of the chromosomes. If we unwind the chromosomes, we have the double helix of DNA. Now, this is very important. This is what Mendel discovered, that, that genes are important. He didn't call them genes, but we now have a molecular understanding of what's going on. And I want to explain this to you, and then give you a couple of animations about what's happening. This is DNA. It makes RNA, a copy of it, and then the RNA is turned into protein. You'll be familiar with this. It's a central dogma. And the order of nucleotide sequence in the DNA determines the order of the RNA sequence, and that acts as a code which determines the type of amino acids that are incorporated into a protein and the type and the order in which they appear. And as I shall explain after lunch, that's crucial for all the activities that go on in life. So let's just get a sense of what actually happens in the process of transcription. And this is an animation done by Cold Spring Harbor, by the way. And, no. I'm not very good at this. It's this one I want, right? Forgive me, I'm actually a computer dinosaur, so I'll make more. Yeah, this is it. Okay, very good. This is DNA, and all these things here are proteins assembling on it to make something called RNA polymerase, which is going now to transcribe this DNA. So it's a big, complicated set of proteins. Do you see that thing buzzing off to the left there? You'll see it coming there. Right, yeah. And on it is this yellow stuff coming out behind, and that's the RNA. You can see it's being copied from the DNA, coming spooling out here. Do you see it? See it spooling out here, because this RNA polymerase is copying the DNA and making that yellow stuff, which is RNA. And in a moment or two, you'll see the RNA released from the polymerase and the DNA to be released into the nucleus. I think that may be happening about now. Look at it. Wait for it. There. It's gone. Okay? This is the process of transcription. And then that RNA is translated into protein, because both of these are nucleic acids. And that translated, the RNA is translated into protein. And hopefully, yeah, here we are. Now, see, there's the RNA swimming out of the nucleus. This is the nucleus. And it's going to, a ribosome is now going to find it. That's this big blue blob. That attaches to the RNA, and another one goes on. And these are all tRNA molecules. You may know about these guys with amino acids on them, all right? And can you see this little red thing beginning to come out here? Yeah? Got that? Oh, it's gone. You'll see it again. Just have patience. This is tRNA with the red blob is an amino acid there. See that guy there? That's the amino acid. And we'll go. Right. Now, this is your ribosome. This is the RNA, and this is the protein coming out of another place being made. This is all animated to show the whole process here. We can see the tRNA is coming in here, putting the protein on, and you'll see a little red thing coming out. See it? Whether it really looks like this, I'm not entirely certain, but this is what they imagine. Okay? So those are the processes of transcription and translation. So this is my second idea, and you're going to get one more idea before you can go and have your pizzas. Okay? And this second idea of the gene is it's the basis of heredity, so very crucial. It explains what I call here the genotype to phenotype. This is the sort of thing biologists sort of talk about. Genotype is the genetic basis. Phenotype is what we actually look like. So we have our genes, that's the genotype. Phenotype is what we actually are. And going through that process of transcription and translation, you move from genotype to phenotype. But also, there's implications for what we are. And this is really a very important issue for all of us, because all of us are partly, at least, a consequence of our own genes. And the question is, how much are we, both physically and mentally, and how we behave, a consequence of what we inherited from mum and dad and what we've been exposed to in our environment? And this is really a raging question. Most of the time, if you say 50% comes from one and 50% comes from the other, in environment and genes, you'll be in the right order often. So there's a contribution from environment, there's a contribution from genes. But it makes us sort of think, how much do we have of free will? If we are determined by our own genes, how much can we be responsible for our own actions? People don't have answers to these questions, but you should think about them. If you've inherited things and you can't do anything about it, should you be responsible for certain of your actions? This is something that we don't quite know, frankly, how to deal with. A second thing that's really important about genes concerns health and disease, because the genes that you inherit from your mother and your father will help predispose you to different diseases. And some of us in this room will be more likely to get certain cancers or heart disease or neurodegeneration. We don't yet know enough to be able to predict that, but we know that's going to be true in principle. So sometime in the future, almost certainly in your lifetimes, perhaps even in the next decade, you will be able, and your children will be able to have, what would be something like a sort of genetic chip, sequence of your DNA that will allow you to predict the likelihood of you getting certain diseases. And that would allow you, in principle, and again I'm only talking in principle, to be able to say, right, if I have this particular genotype, this genetic makeup, maybe I should have this particular lifestyle to be particularly healthy. I'll give you an example. It may be you have a predisposition to skin cancer, and therefore it's absolutely crucial for you not to be in the sun. And whereas others, it may not make much difference. I just give you that as an obvious example, but you could imagine that across the whole spectrum of diseases. And so you could be given a lifestyle that would give you a longer and healthier life. This is a very interesting and very real possibility. But there's another consequence from that. Is such a system compatible with private health insurance? Because remember, private health insurance just assumes that the likelihood of disease is equal across the whole population. That's what it relies upon. But if we know that people are not equal in this respect, then you start to worry about a private health system, because you either have to keep that data totally secure, which I suspect in the long run won't be possible because it's going to impinge on all our lives, or we have to contemplate moving from a private health system to a social-based health system. That is, you treat everyone according to their need, which is rather than their insurance system. That's a fundamentally different way of dealing with health than we have, for example, in the US. So science, I told you, was revolutionary. It really is revolutionary. I think in 50 years, the US will no longer have a private health system based on insurance. It will have to move to a socially based system. And a third thing about genes and genetics that is interesting and relevant today is what it tells us about populations. Interestingly, despite the tremendous variety, apparently, in different human populations across the globe, we are all remarkably similar. Remarkably similar. And so when people talk about race and different distinctions between races, often it's very, very difficult when you analyse those populations to see the differences. We are understanding what we are much, much better as a consequence of the gene and the consequence of genetics. My third idea before lunch, and this is the one which you're probably very familiar with, evolution by natural selection. And if you go to Barnes & Nobles and browse the science books shelves, this is the topic you'll find most books about. And there's two notions in this idea. One is that life evolves, and the second is a major mechanism is natural selection. So where did this idea come from? Well, there's two ideas here. One is that life evolves, and the second is the mechanism of natural selection. Now, the idea that life evolves is actually quite an old one. Some even interpret writings of Aristotle as saying that there was some sort of evolution in the Greek philosophers. But it was certainly discussed a great deal in the 18th century by a number of scientists, chief amongst which was this fat gentleman here on the left, who was Erasmus Darwin. Now, you'll be familiar with Charles Darwin, I'm sure you will be, but you may not be familiar with Erasmus Darwin. Erasmus Darwin was Charles's grandfather. And I'm going to tell you a little bit about Erasmus, because you probably don't know him so well, and he's a charming eccentric, okay? Erasmus was a doctor in England, in a place in the Midlands called Lichfield and Derby, and he was a member of something called the Lunar Society. And the Lunar Society was a collection of scientists and technologists that met once a month at the time of the full moon, because they could ride back on their horses at the end of their discussions under the light of the moon. And they had the nickname amongst their friends, and perhaps not their friends, of the lunatics, because they did everything according to the lunar cycle. Erasmus Darwin had a very serious stutter, but was thought to be the most charming man in the Midlands. And indeed, he married the richest and most beautiful widow in there later in his life. He liked his food, and I've seen his dining table. He had such a fat stomach, he cut out a semicircle from the dining table so he could get nearer to his food. Now, he was a doctor, but doctors in the 18th century really didn't have a huge amount of success. But he was famous, particularly for one characteristic, and that was he was very good at predicting whether you would die or not in your particular disease. And we don't think about this so much now, but that was very important because it meant you could make sure your will was right, you could say goodbye to all your friends, etc. And people respected that. They liked that, the fact that he could predict that. He was asked to become the physician of George III. Do you remember George III? He was the one that lost the US colonies. Remember that one? Okay. He was asked to be his personal doctor, but actually he was a bit of a Republican, and he declined to become George III's personal doctor. He was also characterized by the fact that he would charge patients according to what they earned. Okay? And so most of the charging was with his rich patients, and he treated the poor patients actually for nothing. And this had a bit of a problem for him because he lived in a cathedral close in Lichfield. And he believed in evolution, and he had a motto on his coach which said, everything from shells. Because shells, mollusks, form the sort of blobs. And he thought everything evolved from sort of formless blobs like that. But the dean of the cathedral didn't like it, okay? Thought it was anti-religious. And so told all his rich patients not to use him as a doctor. So he had to paint out his motto so that he could keep his rich patients. He also had other odd things, actually, about him. He was a poet. And at his life, when he was alive, he was better known as a poet than a scientist. I've actually got a book of his poetry. I forgot to bring it back down. I will bring that down a little later. But I'll bring that down after the break. And finally, he was a great advocate for education for girls and set up, believe it or not, the first public girls' school in England in the 1790s. And he wrote a book on education for girls, the first two teachers were his daughters. They were both illegitimate, actually, but both his daughters were the first teachers. So what an extraordinary sort of person. But he argued that life evolved from simple organisms like mollusks, but he didn't really accumulate much evidence for it. It was more a sort of idea rather than something that there was much evidence for. And it took the man on the right, his grandson, who was, of course, very familiar with Erasmus, Charles Darwin, to actually put, firstly, much better evidence in favour of the idea of evolution, and secondly, to come up with a mechanism. And what Charles did was to go on, and I'm sure you're familiar with this, a very famous trip in a little boat called the Beagle round the world, took 5 years, went right round the world, collected lots of specimens, And during those 5 years, he thought about the relationship of the different animals and plants he saw. And he came up in the years after he came back, when he settled down to live in the country outside London, he came up with the idea that all living things were related by descent. And he drew this, the first tree of life, to show it. And it's in this book here, The Origin of Species, this was only 3 months after the first edition. This book's from 1860, and he published in 1859. And this showed, you can see this sort of tree of life here, that everything came from, like we have A here, and it evolved into all these different sorts up here. These went extinct, and these were the species that were, in fact, living today. Well, he thought about this for many years, and the first records we have of this idea was in the 1840s, but he only published it in 1858, when a second scientist called Alfred Wallace had exactly the same idea of evolution by natural selection, which I'm going to describe to you in a moment. But both of these were very courteous people. So Alfred Wallace wrote a letter to Charles Darwin saying, this is my idea about how evolution took place through natural selection. Charles realized it was exactly the same idea as his, but he said that Alfred should publish it before him, because Alfred had sent him the letter. And Charles's friends wrote to Alfred and said, look, Charles has been thinking about this for the last 15 years. You should publish it together. And so they published together in the Linnaean Society in 1858 2 short papers describing the mechanism. And in the following year, in 1859, Charles wrote this 500-page book called The Origin of Species, where he put all his observations from the previous 25 years into that one book. Now, what is the idea of evolution by natural selection? Well, first of all, let's just look at the consequence of it. The famous Darwin's Galapagos beaks. He found that on the Galapagos Islands, there were a load of different finches, and they were all rather closely related, but the beaks were very different, ranging from this big beak to this small beak. And he saw that this beak was used for cracking nuts, this one for catching insects in the bark of a tree. And we put here on the right here different sort of pincers to show how they're adapted for different purposes. And he came to the conclusion that these beaks were adapted to the environment within which those finches worked and lived and how they ate. So let's just tell you about what natural selection is. And then when I've done that, I'm going to combine this idea with the 2 previous ones in a sort of what I hope you'll find an interesting way. So the idea of natural selection is that within a species, within a population of a particular species, there are variants, slight differences, and that these are due to differences in the genes. So there's a range of genetic variation, and that gives rise to a variation in the phenotype, like the sizes of the beaks. Now, those variants which are most successful, that is, those that are best suited for the environment and lifestyle in which those animals or plants are living, tend to reproduce more efficiently. And as a consequence of that, they hand on more of the genes that are important to the next generation. So as a consequence of that, those genes that are important, for example, for cracking nuts, tend to spread throughout the population. And over time, that population will gradually change, becoming more suited to the environment in which it finds itself. And you will see that, for example, the beak might change from being like this to being like this. And it's these changes which eventually lead to different species, and it's this which is the basis of evolution. Now, that's the classic sort of description of evolution by natural selection. Now, let me link it to the first 2 ideas that I talked about, the idea of the cell and the idea of the gene. Whoops. There. Now, it was argued, actually 30 years ago, by somebody called Muller. He argued that living things have properties which allow them to undergo natural selection and therefore to evolve. And he listed the following properties. Life must reproduce. Okay, we all know we reproduce. Life has a hereditary system whereby information defining the characteristics of an organism is inherited during reproduction, that is genes. And thirdly, that hereditary system must exhibit variability upon which selection works. Now, if you have those three characteristics, natural selection is the consequence. It has to be the consequence. And the reason I stated it like that is because I want to take you back to the idea of the cell and the gene to show how they are all linked. Imagine this as a single-celled organism here with DNA, with chromosomes in there, which determine the characteristics of that cell. When this cell divides, and remember this is the basic unit of life, then you'll get two cells which determine the properties of these two divided cells. If there's a mutation or some other alteration in this DNA, then we will get a cell out which will differ from the original parent cell. If this cell is more advantageous in the environment in which it finds itself, let's say it can use sugar better, then this will tend to dominate in the population over succeeding generations. So if you combine the idea of the cell as the basic unit of life and genes as the basis of heredity, together with variability, then natural selection has to occur, and evolution by natural selection is as a consequence. So these three ideas are all linked. They're not separate. They are all linked, and they're all very important to the basis of biology. Well, this was Charles Darwin's contribution, and in The Origin of Species there, he ends this account with his rather wonderful sentence. He got old, as you see. And he said this, I'm just going to read it, so it's so nice. Whilst this planet has gone on cycling on according to the fixed law of gravity, once again, attracted to the law of physics here, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Because he looked upon his evolution by natural selection as a law, rather like Newton's law of gravitation. So let me just finish this particular lecture with this evolution by natural selection. This is the third idea. All life evolves and is related by descent. Natural selection is a major mechanism. By the way, probably not the only mechanism in evolutionary change, but that's a whole other set of lectures as to other things that can influence the change of including genetic drift and the like, but a major mechanism in evolution. And then there's two other ideas that I want to sort of leave with you before lunch. The last one is simple. If we are all related by descent, in a very fundamental way, that means we are all related to every living thing around you. Every plant and every animal has some relationship with you. Now, if we are the most advanced beings on this planet, do you not think we should have some responsibility as stewards for the rest of our relatives, that is, all the other living things that are around us? And I think evolution by descent and relationship by descent, by the way, there's an S in there which has been left out. It's not decent. Well, it is decent, but I mean it's also by descent. Then we should be stewards for our relatives. And I think it's a very fundamental argument as to why we should take responsibility for the biosphere. So that's just a sort of one idea for you. The second idea, which is interesting and something you might want to read about, is the notion of kin selection. And that may form, it's certainly relevant to altruism and morality. Kin selection, which was a notion that came from a scientist called Bill Hamilton in the 60s, was the sense that you feel a responsibility for your relatives because they share the same genetic makeup, and that you are nice to your relatives and your kin as a consequence of that. And some have argued that out of that, ideas of morality can emerge. Once again, this is a complicated issue which is important for thinking about, and there's no clear view on this, but kin selection can form the basis, in part, for thinking about altruism and morality. And if you're interested in that, there's many books that are written about that where you can find out more. Well, that brings me to the end of this lecture. In part 2, I'm going to think about two ideas. This will be a little shorter than part 1 because I thought you'll be getting a bit more tired this afternoon, okay? I'm assuming you're tough in the morning, okay? And I'm going to talk about two ideas. One is life as chemistry, and a second developing idea, which is life as a system. So I hope to see you after lunch. Thank you very much. Thank you. I forgot to bring a book out this morning. I told you Erasmus Darwin was a poet. I don't know if I emphasized that he wrote all his scientific theories in poetry. And I have a book here from 1790. And let's see, look. This is called The Loves of Plants, this poem. And it's all about, plants reproducing and stuff. Okay, so I gave you 3 ideas this morning, and this afternoon it'll be a little bit shorter, so you can take lots of deep breaths. You don't have to keep going quite so long as you did this morning. And then we're going to have questions afterwards, which can be on both this morning's and this afternoon's talk. In part 2, I'm going to develop on the 3 ideas that I told you about: the cell, the gene, and evolution by natural selection. And I'm going to talk about life as chemistry, and then a new idea, which is possibly not a real idea of biology, but might be. We'll just see, okay? But the first idea really is that's life as chemistry. So what am I talking about here with life as chemistry? Really, this goes back to Aristotle again. You remember Aristotle? I mentioned him this morning, Greek philosopher of the 4th century BC. And he was trying to understand how living things worked. And he proposed that life, living organisms, had a sort of a natural end, a goal, and that they were driven by an inner vital force towards that goal, which he called vitalism. And this idea of vitalism dominated biological science generally, for many, many centuries. And the notion was there was something special about living things that somehow separated them from the laws of physics and chemistry, which was a sort of vital spark, something that was sort of different. Now, there are different things about biology, which I'm going to talk about this afternoon, but they are still subject to the laws of physics and chemistry, whereas the idea from Aristotle was that perhaps that wasn't quite the case. And I'm going to tell you how the idea that life is chemistry was actually brought about, and how we got to the position where chemistry and physics are seen as the mechanistic basis of life. Now, would you believe that the origins for this idea start with wine? It's not where you'd think that it would start, but it starts with wine. And if it starts with wine, you can well imagine that the French are not very far away from it. And that is the case here. And I'm going to tell you about a very famous scientist, really the father of modern chemistry, called Lavoisier. And this is a picture of Lavoisier, which you can go and see in the Metropolitan Museum of Art. But actually, this picture used to belong to Rockefeller University. In fact, it used to hang in the office that I have here. It's a gigantic canvas. And this is Antoine Lavoisier. So let me tell you a bit about what he did. Well, he built on the ideas of the alchemists, who were interested in, of course, making gold, as many of you know, but they were also interested in how burning water was made. That's alcohol. And they were particularly interested in how burning water was made during fermentation. And Lavoisier made a study of fermentation. Fermentation, of course, is where you take fruit extracts like grapes and you put them into conditions where they make alcohol. And what Lavoisier did was he took a grape extract, he measured the sugar content in it, and he found that during fermentation it was mostly turned into ethanol. And he noted that you had something called yeast in there, a sort of living organism, but that didn't seem to change. It was there at the beginning, it was there at the end. But there was a conversion from sugar into alcohol, and yeast seemed to have something to do with it. Now, Lavoisier didn't go much further than that, because as well as being a great scientist, he had one other job as well, which was actually less helpful to him. He was a tax collector, and he was a tax collector for the king, and he made a mistake of being a tax collector for the king just before the French Revolution. And at the French Revolution, he was guillotined for being a tax collector, which is actually, a great tragedy, of course. He lost his head in 1794, and his judge, I'm going to read out what his judge said when he was judged. He said, the Republic, the French Republic, has no need of chemists and savants, savants being wise people, and later in the day, he lost his head at the guillotine. You can see the guillotine over here. So he didn't get very much further, obviously, with this research. And we needed another Frenchman, also interested in fermentation and wine, called Louis Pasteur, to take it further. Now, Louis Pasteur was originally a chemist, and he was working in northern France, in Lille, and he had a businessman come to see him, whose job it was to make alcohol out of sugar beet. That was his job. He fermented sugar beet, okay? And they noticed that sometimes the fermentations didn't work. And they wanted to know why. And Pasteur turned his microscope to looking at the fermentation, and he discovered the following very interesting fact. He found that when the sugar beet, which is making sugar, is turned into alcohol, there was yeast in the fermentation. When it went bad and you made a sort of vinegary acid stuff, then you didn't see yeast, you saw bacteria. Okay? So he linked, in a stronger way than Lavoisier, the yeast and the bacteria to the chemical transformations going on in the fermentation. And he argued, and I'm going to quote, that fermentation was a physiological act, that is, an act of a living thing, yielding chemical products necessary for the cell, the yeast cell or the bacterial cell. And in this way, he helped cure the problem that the businessman had, because he said, if you're going to get a good fermentation, you have to put yeast in there. But he was also a good scientist as well, and he made the following conclusion, that such chemical reactions, the one that are going on in fermentation, are an expression of the life of the cell. Now, it doesn't sound a very important sentence, but it is. I'm going to say it again. Such chemical reactions are an expression of the life of the cell. He said it in French, of course, by the way. And what he was arguing was that living organisms, like the yeast cells, like the bacterial cells, were making chemical reactions, and that was an expression of life's processes. So this was really the first very clear statement that chemistry is very closely connected with life. We have to go on a little bit longer. Notice we're once again in the 19th century. I seem to speak nothing about anything else but the 19th century. We had that with the gene and with evolution of natural selection. We have this as well. The Buchner brothers then went on and they took yeast in the late 19th century and they broke it open. They smashed up the yeast cells and they extracted from the yeast cells a substance which they called zymase, we now call enzymes, which could make the alcohol in a test tube, in vitro, without the yeast. So put all these ideas together, and by the turn of the 20th century, last century, it was argued that life's processes could be reduced to chemistry. Chemistry was the basis of life. And this was the birth of biochemistry, which dominated so much of the 20th century. In 1910, and I'm going to refer back to Rockefeller again, there was a scientist here called Jacques Loeb. And Jacques Loeb stated that, in his view, the cell should be considered as a chemical machine. And that's exactly where most of this afternoon's lecture is going to be talking about, the cell as a chemical machine. Now, that's the origins of the idea. Now, how does it connect with what I told you about this morning? Well, the the zymase, that is the little chemical machines that are in cells that made the alcohol, these are proteins. I'm going to tell you about proteins in a moment. And these proteins are made by the genes that make up the hereditary material. So we can connect that biochemistry to the genetics that I told you about this morning. Now, you've all come across proteins and enzymes, I know that, but let's just again try and sort of view exactly what these amazing things are. This is a space-filling model of a protein, and each of those sort of little balls in here is an amino acid, because I'll remind you that a protein is a string of amino acids. And proteins are enzymes. They undertake chemical transformations in the cell. And the way they can do that is because they have particular shapes, and the particular shapes have different charges and other chemical properties on them. So in looking at this structure here, you see it has a particular shape which is determined by the sequence of the amino acids that make up the protein. And the ones in purple, for example, will have a particular charge. So you have a positive or negative charges distributed around the surface of the protein. And you'll also have surfaces of the protein which like fats and other surfaces which like water. And all of this makes it a very specialized chemical environment. And these very specialized chemical environments can do all sorts of amazing chemical and physical transformations within the cell. Now, I'll give you an example. This is a structure of Rod McKinnon. Rod McKinnon got the Nobel Prize in 2003, and he's there in the audience, together with his nephew, actually, I think, who's just sitting next to him too. And Rod McKinnon, did that rotate then, just for a bit? Shall we do it again so I can... No, why doesn't it rotate? Yeah, there we go. Very good. And what this is, and this is what Rod solved the structure for, this is a protein which can carry ions from the outside of a cell to the inside of a cell. And this is an exquisite little chemical machine that can transfer these ions from one side to another. It's absolutely crucial for understanding how nerves work, so it's really, really important. And it just shows you the sorts of things that proteins can do, in this case acting as an ion channel. And it's all due to the shape of the proteins, the particular shape, the distribution of the different types of amino acids that give different charge distributions and other properties. So what you can get, and I tried to emphasize this by, these are all different enzymes which have got different shapes in them, and I'm trying to sort of draw this as in a sort of metaphorical sort of way, and it's really changing the shape of the molecules that enzymes are actually doing. So they can bend chemicals, so to break bonds and reform bonds. And the way to view this is that there's lots and lots of different chemical reactions going on in the cell. Now, that's interesting enough, but there's something else that makes this even more interesting, and that is that within a cell, there's many, many hundreds, if not thousands, of chemical reactions going on at exactly the same time. And I've got up here a drawing of the map of metabolism. You don't have to sort of understand what this is, except that each of these dots is an enzymatic reaction, changing one chemical to another chemical. They're all linked together in a very complicated way. And even on this very limited part of what's going on in a cell, there are hundreds of different reactions. Now, what you have to understand is, in each cell, which is only a few micrometers across, that's a millionth of a meter, there are thousands of chemical reactions going on simultaneously. Now, how is that possible? Well, it's partly possible because each of the different enzymes, this is one way to view it, makes a very specialized chemical microenvironment. In other words, the structure here undergoes a certain chemical transformation which is different to the structure here. So you have compartmentation down at the level of the molecules of the enzymes. But that compartmentation is seen all the way up, even into the structure of the cell. And this isn't normally the way that you're taught about cells, but it's the way I want you to think about them. This is a diagram of a cell. This is a nucleus. These are mitochondria. there's different other structures in them. I want you to think about this as many, many hundreds of different compartments where different chemistries can go on simultaneously. It's a different way of thinking about it. Within a cell, there are hundreds of chemical reactions going on at the same time, and part of the structure of the cell is to bring about that compartmentation of chemical reactions. And you do that with having different membrane components like the nucleus, having complexes of proteins together, having single proteins themselves, all giving individual little compartments. So the cell's complexity is there because you need complexity in the chemical microenvironments. So let's now put all this together. We have a gene, the hereditary material. The gene encodes proteins. Many of those proteins are enzymes carrying out chemical reactions. Some of them are structural, but many of them are these little chemical machines. Put all this together and we get the behavior of the cell. The cell, remember, the basic unit of life with the functions of life. Put the cells together, we have a tissue. Put the tissues together, we have organs and an organism. And you can see how all the ideas I'm talking about are linked to give you a sort of explanation of biology. So where has this idea taken us to? Well, first of all, life as chemistry destroys vitalism. And the notion that there's some special forces that we don't understand is eliminated. We can understand how living things work in terms of physics and chemistry. Very sophisticated physics and chemistry, as I shall talk about in my final idea, but even so, standard physics and chemistry. But it's also very important for sort of mechanistic explanations of many diseases and for the basis of the pharmaceutical industry. Now, why do I say that? That is because one of the most powerful ways of understanding disease is that sometimes this chemistry goes wrong. In certain cases, that's very simple. There's certain diseases where you can't make an amino acid, such as phenylalanine, and that will give rise to a whole set of diseases because you can't make that one amino acid. That's simply understood because there's one enzyme which is defective in the human beings that suffer from that disease. Diabetes is another disease where you have an error, a mistake in the chemistry that is going on. So we can understand many diseases in terms of chemistry. And what that also means is that we can try and correct that chemistry with drugs, with intervention. And that's the basis of the pharmaceutical industry. If you think about it, the pharmaceutical industry is making drugs that interfere with specific chemical reactions in cells and tissues and organs that change the chemistry that's going on such that you can try and correct the disease or the ailment that you're suffering from. Traditionally, many of those drugs that have been developed by the pharmaceutical industry have come from folklore. And they've just sort of purified the chemicals involved. I'll give you an example. Aspirin, which many of you will know about. It is called aspirin here, isn't it? Or is there another? Yeah. Thank you. I sometimes get confused, you know. What continent am I on? Anyway, that was discovered because people knew that they could help toothache if they chewed willow bark. It's really true. And that was because there was a lot of aspirin in the willow bark, and then the German pharmaceutical industry essentially purified aspirin from that source, and then they make it chemically. So some of the pharmaceutical industry is derived from, if you like, folklore. Other, increasingly, is built on knowledge of all the chemistry that goes on in cells and trying to intervene with it. One very famous drug you may have read about is, for example, Gleevec. This is an anti-cancer drug which is used to treat certain leukemias. And Gleevec interferes with a chemical reaction that induces cancer in particular types of leukemias. And that can actually have very significant effects. This is sort of designer drugs which will have very specific effects in dealing with that particular disease. So life as chemistry is both important for understanding disease and is central to medicine and the pharmaceutical industry. So it's really key. But this is a sort of introduction, really, to my final idea, which I want to spend a little time on, because it's one which you'll be less sort of familiar with. And that's built on the idea that life is chemistry, but a rather special sort of chemistry. I've already hinted at it. It's very complicated. We have so many chemical reactions going on in the cell at the same time that we've got to think about how is this all organized together. So with this fifth idea, which is biology as an organized system, this is a new and developing idea, and maybe it won't reach the same status as the ones I've talked about up until now, but I'd like to finish these two lectures discussing this as a possibility for thinking about the future. And the idea here is that living organisms, let's say cells, are very complicated, organized systems built on chemistry and physics, but which are organized so intimately that they have properties which are difficult to understand, and it becomes a major objective to understanding them in the future. Now, what is this idea? Well, this idea is one that's becoming quite popular in biology, but actually, once again, is quite an old idea. It wasn't one that has been invented in the last few years. It was developed, talked about in the 19th century, as always, and it's based on complex systems and systems theory, which have been developed to better understand the weather, transportation, social networks, and also biology. All of these are complex systems and difficult to predict exactly how they work. And it was developed a great deal in the 1940s and 1950s when the theory behind computers was first being talked about. And sciences like cybernetics were being invented. Cybernetics is how you manage information, for example. Game theory, information theory, and systems analysis all emerged in the 40s and 50s. And what they tended to do was analyze these complex systems and focus on a variety of aspects. Firstly, they're complex, and they'd like to think about the behavior of the whole system together. Sometimes the behavior didn't seem very determinate. That is, you couldn't predict very well what would happen. It had sort of probabilistic aspects to it. The maths that describes it is sometimes chaotic and non-linear. When I mean chaotic, by the way, I don't mean that it simply is incomprehensible. I simply mean that it's maths which is sometimes called on the edge of chaos. You know, if you go a little bit further, it's totally chaotic. But you're not, if you go a little bit further back, it's very determinate and very regular. But you're occupying a sort of space in the maths which is in between something that's very determinate and something that's very chaotic. And it makes it very difficult to predict what's going to happen because very small changes in that area can have huge effects subsequently, and it's difficult to predict that. Have you heard of the metaphor where when people are talking about the weather, they say somewhere in the Amazon jungle, a butterfly flaps its wings, and eventually there's a typhoon in the Japan Sea. Have you heard that? Yeah. And that's exactly what we're talking about. A little fluctuation somewhere leads to dramatic effects later somewhere else, and it's difficult to connect them, but that's because you're operating on the edge of chaos. Now, in biology, this systems approach operates at a variety of different levels. It operates at the level of the cell, which is where we're going to focus today, but it also acts at the level of the organism, operates at the level of behavior, at the level of ecology, and at the level of society, and for that matter, at the level of evolution. And it's possible that rules that we learn from study at one level will also help us with studying at another level, because biology as a whole is a very complex system. So what's the sort of idea about? Well, specifically, the sort of agenda here, because I think that's the best way to describe this, it's an agenda rather than a proper idea, is the agenda is that a better understanding of biology could be developed if we translate the descriptions of chemical and physical processes that go on in cells and organisms into the behavior of complex organized systems. So you want to take those chemical reactions I was telling you about and see how they all operate together as part of a complex system. And it emphasizes different aspects, like it's a particular emphasis on information, gathering information, processing information, storing it, on feedback controls and network behaviors. And I'm going to tell you a little bit about all of these things. Now, I can see some of you looking a bit glazed, okay? That's because I talked rather a lot there without... Now, if you're getting a bit glazed, it's time for a deep breath, okay? And concentrate again, because I'm going to go a bit concrete again, and it'll be easier, all right? But that was just to give you a bit of a general sense of where we might go. And we're going to go easy because we're going to talk about the double helix, okay? And this is the structure that Watson and Crick discovered, built on the discoveries here I told you this morning about DNA being the hereditary material. And I mentioned at the time that the structure of deoxyribonucleic acid was a double helix. So you can describe this in terms of that chemical structure, okay? It's a double helix. Now, it's beautiful. You know, it's iconic, it's beautiful. But the truth is, it's not beautiful really because it's a beautiful structure, though that is true. It's beautiful for what it tells us about information and heredity. And this is actually the connection I want to emphasize for you. So let me just explain this and say what I mean. The structure of DNA, as I'm sure all of you know, is a string of nucleotides. And they, if you consider this like a ladder with two sort of lines going down, the ladders are connected by, both sides of the ladder are connected by rungs, and these are generated by pairing between the nucleotides that make up each of the sides of the ladder in such a way that, and you don't have to worry about what they are, G always pairs with C and A always pairs with T. That's a very simple structure. But there's a consequence to that which is beautiful. And that consequence is this. First of all, it means that each of these strings here is made up of different letters, of A, G, C, or T. And that means that you have a code for information, right? And think about that. A language, a sentence, is just a series of letters, right? Now, we're used to 26 letters in our alphabet. All right? This is an alphabet with only 4 letters, but you can still write sentences with 4 letters. It doesn't look quite the same as our sentences, but you can still write sentences with 4 letters. Therefore, information is coded in the sequence of letters that make up each of those strands. The second thing that emerges from this structure is that the two strands are related to each other, because I told you that G always pairs with C and A always pairs with T. So that means you have a complementary strand on the other side of the molecule. So if you unwind this molecule, then when you reform another double chain, you will actually make a precise copies of the original one. Do you get that? Okay? So that's the second characteristic. Not only do you have information encoded in this molecule, but you can copy it with a simple chemical reaction. Now, let's now draw the general principle. What we're saying here is that the chemistry can be best understood in terms of biology by focusing on the information and how that information is replicated. And this is, if you like, a higher systems property. And what you've done is you've moved from a chemical description to a description that's relevant to the biology. Did you get all that? Yes? Sort of. Well, just remember one thing. The chemistry is responsible, ultimately, for the information here, and understanding the chemistry helps you understand the information. Okay? And that's crucial to what we're doing. Let's try another example. Do you know what this is? No. I took a picture of this. It's a governor on a steam engine, and it was on a boat in New Zealand that I was travelling on 18 months ago. And it's a very clever device, and because it was invented in the 19th century, we can understand it, you know? Because we can never understand anything invented after about 1950. And shall I tell you what it is? It sits on top of a steam engine. Now, this is relevant to my talk, if you think I'm losing my marbles, by the way, okay? And the steam engine is letting steam into the engine, and it's rotating things, okay? In this case, the engine driving the boat. As this shaft rotates faster, these balls get driven out by centrifugal force. You got that? Yeah? As they are driven out, this is lifted up. Do you see? And that then touches this, and this is a valve which controls how much steam goes into the engine, right? Therefore, the faster the engine goes, the balls go out, and it shuts off the steam going into the engine, and the engine slows down. And this is why it's called a governor, which governs the speed of the engine. It keeps it constant, you know, okay? And this is a negative feedback loop, right? And why a negative feedback loop? What I mean is that as the engine goes faster, it feeds back on the mechanism driving the engine in a negative way to slow it down. And that leads to what we call homeostasis, fancy word for meaning keeping things the same. And this is central, this idea, to the whole thing that I'm talking about. Because such feedback loops control the chemistry that's going on in cells. Here's a negative feedback loop, okay? I want you to imagine that you've got 3 chemicals here, and you've got an enzyme here and an enzyme here converting A to B and B to C. And the notion is that when C builds up, it feeds back in a negative way on the enzyme between A and B, slowing that reaction down, slowing the amount of C that is made, and therefore controlling the overall rate of flux through that pathway. Got it? Just like the governor on the steam engine, okay? As this gets faster, then you have a built-in mechanism that slows it down. And this controls how chemical reactions go on in the cell. This is a negative feedback which keeps things homeostatic, that is the same. But you can also build different sorts of feedback loops which actually act as switches and switch things on. And you'll see an example here with a positive feedback loop. Same system, A to B to C. There. Dance of the Seven Veils. You're not allowed to look at that yet, okay? Positive feedback loop. You're going from A to B and B to C. But now C speeds up the reaction of A to B. What's the consequence of that? What it means is, as soon as you get just a little bit of C, you speed up this reaction so you get lots of C. It's a way like switching on a light switch, on or off. As soon as something starts to happen, it goes all the way. You're not sort of dithering around with the light half on and half off. You just go for it, okay? That's called positive feedback. Biology is built on feedback loops like this. The chemistry that's going on is built up of these sorts of feedback loops and other sorts of ways of handling information. You don't have to read this. This is a very famous chemical circuit in bacteria, worked on by Jacob and Monod in France in the 50s and 60s, called the lac operon. And this is a negative feedback loop controlling the production of an enzyme important for lactose utilization, built entirely like that negative feedback loop there. And isn't it interesting? They discovered this in the 50s and 60s, just after cybernetics and information processing was being talked about. And this tells us something else. Real scientific advances are often made in between subjects. This is biology, but it was built on theory that came from people thinking about computers and information. And if you just stay in your own little silo, you never discover anything. You have to be looking out and sort of interacting with other areas. Okay, so that is one part of what we're talking about. We have to understand the systems and the regulation that's going on. And these sort of feedback loops can produce switches and timers and oscillators and all sorts of other things that go on in cells. And we have to understand how that works. Now, there's another aspect to this that I want to introduce you to, which is the fact that all this chemistry is connected. Okay? Now, I showed you that with that metabolism map, but I want to use another analogy that may help you. And that is the analogy of an electronic circuit. Okay? And what this is, I don't know what the hell this is actually, but it's probably... this is probably a computer chip or something like that, okay? But the main thing is, it's connected by all these devices, you know? They're all connected to each other to make a network, right? And the consequence of this is either it's a computer or it's a radio or a television or something, which, if you like, the electronic components generate higher level behaviors. Okay? Got that? The electronic components generate higher level behaviors, which are important. So we have to think about circuits. And we have to think about these circuits in biology. So this is one way of doing it. This is another thing. I think this is a radio, by the way. It looks simpler than the other one, doesn't it? And each of these are different genes, proteins, that have been made by different genes. And what somebody's trying to do is think, look, actually, these are all different proteins in a cell, and they're connected in different ways, and they are generating behaviors. This is generating a radio signal. In the cell, they'll be generating switches that switch things on and off or communicate or whatever they're doing. And we've got to use the metaphor of the electronic signal to work out what's going on. Now, circuits are interesting. Networks are interesting. And we don't understand networks. But we can help, different areas can help each other. Do you know what this is? Yeah. Well, I'm glad you can all read still after lunch. Exactly. It's an airplane route hubs. And it's the US, and what it says here is that we have 2 or 3 places, and in this particular case it looks like New York, Atlanta, and Cleveland or somewhere, yeah? That is Cleveland, isn't it? Somewhere, yeah, I don't know. Anyway, and these are hubs for a particular airline. I don't know which one it is. Anybody know? Continental, thank you, great. Must be a scientist who travels a lot, I'd say. Now, what I'm trying to point out here is the following, that many of these places are connected. But if you wanted to get from, let's say, here to over here, then what you would do is you would fly to here, then you'd fly down to here, then you'd fly over to there, let's say, okay? And the people who analyze all of this say the most efficient way to get good flow throughout the whole of the country is to have a few hubs where most of the flights come into. So you fly the short distance flights, you go into a hub, and then other short or longer distance flights go out from there, and then you go to another hub and you go out from there. And you can organize it all so the whole thing works. Now, what's really interesting is you can describe this by the, if you like, the numbers of connections that a hub has with many places versus, say, a place like this that isn't a hub, okay? Now, and you can get a mathematical description of that. If you now look inside a cell and you look at the interactions between proteins within a cell, you can describe them in much the same way. In other words, there are certain hubs, certain proteins, certain reactions, which are connected everywhere in the cell, and there's others which only have very, very local connections, if any at all. And there's something interesting here that we don't understand. Not yet, but we will do. And what this is sort of telling us that maybe the rules that you can learn about transportation networks may be helpful in understanding the rules that go on in cells. I told you this is a developing idea. We don't know that yet, but there's something interesting there about how all this works. And if you're interested in this, this is called a scale-free network, if anybody wants to sort of go further in this sort of stuff. So there's things to be understood here which we don't know about, and they are to be understood from comparing different systems in such as airplane networks as well as cells. Now, let's look at this, networks and pathways, okay? This is the way that biologists traditionally think about things, called a linear pathway. You know, we start here and we go boom, 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 and we end up here, okay? This is actually the way, traditionally, that people think. You know, we're pretty linear. You know, we do something, go blah, 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 okay? That's sort of the strategy that, well, I'm going to elaborate on this in a moment, but it's a strategy that we quite often use in thinking about things. But the reality is, for reasons that are becoming clear, is that the networks that go on in biology are much more complicated, you know? So in other words, instead of being a linear pathway like this, you'll have crosstalk from one pathway to another. You'll have some things inhibiting, some things activating. You'll have redundant pathways. It's a mess, okay? And it's a mess for various reasons, one of which I'll tell you about in a few minutes. So we've got to move away from the linear pathway to complex networks and thinking about complex networks. But there's implications of that. okay? And one of them I've already hinted to. We tend to think like this, or at least ways men tend to think like this. Now, it may be that more complicated ways of thinking about things are a much better way to contemplate these things, and this sort of manly approach may not be the most useful way to think about biology. And I have a metaphor for this as well. And I want to suggest to you that, whoops, I've done it again. I want to suggest to you that we've been too long in this way of thinking, and we should start thinking more like this, okay? Because this is complicated stuff. So that's one thing I want to say about networks in biology. There's a second thing, which is quite interesting and unexpected. Now, imagine that we do have a simple linear pathway, okay? Imagine this as a signaling pathway. That is to say, what can we communicate through this pathway? Well, a simple-minded approach would say, well, you can only really communicate a yes and a no signal. You know, imagine this is you press a sort of a key here, and you either have a light or you don't have a light, okay? Or you either have a noise or not a noise. Here's one. Oh. Can I have noise, please? This is so... Right. Every time I put my finger, can you see my finger? Right. What I'm doing is I'm starting there. And that noise is coming out there. And so you either hear a noise or you don't. Right? You got that? Now, I want you to imagine the same pathway. I'm going to say something interesting now, actually. Imagine the same pathway, but now introduce dynamics. What do I mean by dynamics? I mean changes in time, okay? So instead of it just being an on-off signal, what you do is you have changes in time. I've tried to draw that up here, where you could have the same signal, but instead of having a constant signal, you have a pulse, and you have a pulse like this and this, or you have pulses like this, this, and this. And that means different things, okay? Now, let's go to a metaphor again. The metaphor is the Morse code. You all heard of the Morse code? And there's a statue to Samuel Morse in Central Park, if you want to look at it. Now, this is the Morse code. You see, we've got a letter A, letter B, letter C. Have you got it? Yeah? That's all dynamics, right? Now, think what that means. Now you have a language. And with that language, you can now write the works of Shakespeare. I'm going to say that again. With that language, you can write the works of Shakespeare. You move from being an on-off signal to being able to write Shakespeare. Isn't that amazing? And all you've done is introduce dynamics into signaling. Now, we do not know how important dynamics is in signaling in biological systems. But I would be astonished if living cells, tissues, organs, and organisms are not making much more use of dynamics because of the richness of information transmission, and we haven't yet got to grips with it, simply by thinking about it in moving from a red and green signal to writing Shakespeare. It would, I think, be impossible to imagine that it doesn't use dynamics in that sort of way. And this, again, is part of the sort of thinking that I think this new approach has to embrace. Let's think about another property of living things. Living things have shapes, very characteristic shapes. All of you around this room are recognizably human. Well, we all have characteristic shapes. We all are recognizable as a consequence. as being Homo sapiens. And being a particular shape is something that's very characteristic to biology. I've drawn up here a phage, bacteriophage, which has a particular shape. Now, bacteriophage, we understand how their shapes are made. It's all direct chemical interactions. It's molecular-based. It's very, very direct. It's like the shapes of those enzyme molecules I talked about. You know, it's easy to understand. But if you go to a cell or an organ or an organism, it's much less easy to understand because you're actually getting control of space over an extended area, an extended region. Take a cell, micrometers across, but that's far, far bigger than the angstroms that make up molecular distances. There's got to be communication across bigger distances. Micrometers in the case of the cell, millimeters in the case of tissues, centimeters in the case of organs. Knowing how you get spatial order to generate particular shapes and particular sizes and dimensions is frankly something us biologists still don't really understand. And it comes from the chemistry, and it comes from the interactions of molecules, but it generates higher order structures and organization, which we need to understand, and again, may emerge from thinking about the overall system. People have thought about this. A very famous mathematician called Alan Turing, who was also responsible for the theory behind computers, generated equations that showed that chemistry could generate structures on a macro scale. So this is a chemical reaction. It's a chemistry discovered by a couple of Russians. But here you have it coming out in waves with orders every couple of millimeters. Now, this has not been shown to operate very much in biological systems yet, but we have to understand how spatial order is generated. And this is the way we can start thinking about it. I don't think it will be the solution, but it's a way of thinking about it. But biology is complicated. And this is one of the reasons I went back to those networks. You remember when I talked about networks, I said there's redundancies. Remember that? Now I'm going to take you to the idea of evolution by natural selection and take you back to, and these clocks are relevant to it in a moment. Just look at them because they're nice to look at. But listen to me, okay? Now, if you are a designer and you want to design something like a clock, then you think about the problem and you design something. These 4 clocks were all designed by somebody called John Harrison in the 18th century. And he was trying to win a prize from the British Admiralty for inventing a very accurate clock. And Dava Sabel wrote a book about this called Longitude, if you're interested, which, if you could measure time very precisely, allowed you to measure where you were on the globe, okay, the longitude on the globe. Now, you needed to have a very accurate clock that could work on a sailing boat. And this was quite difficult. So he invented a clock, and it was this one over here. And they sent it to the West Indies and back from Europe. And by the time they got back, he was out by 100 miles, okay? So he ditched it. He said, he looked at the clock and he worked out what was wrong with it, and he invented a completely new clock, okay? And he did it again, and this time they still made a mistake, and he did it again, and he invented this clock, designing it from scratch, until eventually he got this clock, which bore no resemblance to the initial clock, right? Now, the point that I'm making here is that these clocks were designed, and these clocks were designed from zero. But that is not the way that life evolves. Life is not designed. I use my words very carefully. Life evolves. That means that if you started with this clock, it would have to evolve through this process. And that means you couldn't just dismantle it and reinvent it and redesign it. You'd still have to have the bits here over here, or at least the remnants of them. Do you see what I'm saying? Therefore, it is very likely that biological systems will be redundant. They will have ways of doing things, which they did here, which will be replaced by more efficient ways, but you still have the residual system in place. So that when you try to analyze it and tease it apart, it actually is quite difficult to do so because you have a very efficient machine here, but if you destroy that by taking a component out, you might reveal the structure over here that's still somehow lurking in the background and may still be operating in part. Now, I use this as a metaphor simply to communicate to you that when you analyze complex biological systems, we have to go right back to our network here, because you might end up with this rather than this, because this has been designed, whereas this has evolved, okay? And that makes this system much more difficult to actually analyze and, for that matter, to understand. Now, what I've done is I've focused on biology as a complex system. And I want to end with two more thoughts. And the first thought is that this sort of thinking, this fifth idea, may be helpful in thinking about life outside Earth. And you'll know that one of the interesting things in astronomy is looking for planets around stars and people thinking about whether there's life there or not. Now, traditionally, when scientists have thought about life on other planets, for example, in the solar system, and here's Mars rover, the rover exploration vehicle that was sent to Mars, they've looked for life by looking for the chemistry of life, looking for amino acids or nucleotides, the components of life. But it may be that life, as a complex system, could be built on different chemistries. Not the chemistries of carbon, which is the basis of amino acids and nucleotides, but other sorts of chemistry. Some people have imagined chemistry built on silicon might produce the complexity necessary for some forms of life. Some people have argued that clay particles, if manipulated in particular ways, could perhaps have certain characteristics of life. And the point I want to make here is only that if we're going to think about life in a sophisticated way, we may want to think about complex systems rather than simply the chemistries, because that may reflect more what life actually, the core things as to what life might be. So that was the one point I wanted to make. And the second point, and actually my last point, is a suggestion that maybe this means that biology is about to undergo a real revolution, because it could be that a complex organized system that underpins cells and tissues and the like is not going to occupy a common sense world. Now, what do I mean by that? I mean this. I'm a research biologist, and when I do experiments, I imagine a picture of a cell, and I imagine chemistries going on in there, and it's really, I'm imagining the world of this room down at the level of a microcosm, where I've got molecules banging into each other, just like you might walk into each other, and there are things happening. I'm reconstructing a common sense picture of a cell from my everyday world. I mean, that's how I think about it. Now, physics used to be like this. Newton imagined a world that is common sense in its basis. So, you've probably been taught billiard ball Newtonian physics, where one ball hits another and there's a reaction. And then two things happened about 100 years ago. The first was Einstein's theory of relativity, about 1900. And that began to sort of mix up space and time, okay? And it's quite difficult to understand. It's like one of these things where you read all these popular books and you think you've got it, and then you close the book and it slips away. Do you know what I mean? You know? And this is because it actually only really makes sense in the mathematics. It doesn't make complete sense in our common sense world because it doesn't occupy a common sense world. But if you think you have trouble with relativity, move on 25 years and try quantum mechanics. And quantum mechanics occupies an Alice in Wonderland sort of world. It's utterly, utterly incomprehensible. Now, I used to be depressed by this because I used to read these books about this thing and I thought I was too stupid to understand them. Then I had the fortune of having a daughter who became a high-energy physicist, and I realized, sitting here actually, I realized she doesn't understand it either. And that's because it doesn't occupy a common sense world. She understands it in terms of the equations, because it makes sense there, but you can't imagine it. You can't imagine Schrödinger's cat, both alive and dead at the same time. It does not occupy a common sense world. Now, the question I ask you, and I don't know the answer, is the complexity of biology that I've talked to in my final idea here, going to move us into a non-common sense world, a world where we don't imagine pictures, as the way I operate, but where we move into more of a world that the physicists have had to inhabit for the last 75 or 100 years, dealing with the extremes of physical reality, the very large and the very small. Maybe the complexity takes us out of our common sense world. And if that's true, then I think we're in for very exciting, though possibly unexpected and strange times in 21st century biology. But I certainly hope it'll be an interesting time. Thank you very much. Thank you. If we're not going to have any questions, that's okay too. Oh, you have one. Could you get to the microphone, because we can't hear you? Well done. Okay, silence for the questioner.
Hello.
Hello.
My question is...
What's your name, by the way?
Oh, my name is Irina. Nice to meet you.
Good to meet you.
My question is about what you spoke about just now with life on other planets and how we have to think about things differently. So would you be willing to say that if we find life on other planets, we might not know that we found it, we might not recognize that it is life because it's, as you said, not part of that common sense world that we think of?
Okay, so the first yes is the answer to the first part, but not quite a common sense argument. You see, the only way we can easily look for life is to look for the different chemistries, looking for certain chemicals like nucleotides or amino acids, or something that life as we know it on this planet is making chemically, because that's the way we can, if you like, probe the so far away. But if life, if there's forms of life which are built on complex systems, built on a different sort of chemistry, then that approach won't find it. So you're right in the first part, we may not recognize it. Just possibly, some people have argued, we have life on this planet we've not recognized because it's not built on carbon. I think that's unlikely, but that's what they have argued. But if you go into another world where life is not very efficient, then maybe less efficient forms of life can survive and we might find them. But we wouldn't know how to recognize them with the ways we have. If we were there and could study them, then that would be different. So, yeah, I think that is possible. It's a good question. Maybe we can't recognize life of that sort. Just go back to them. If you want to ask, just, look, that's it. Do what those... Yeah, we'll go over there. Yeah. Okay. What's your question?
Is this on? Okay.
Yeah, just talk. It'll work.
I was just wondering what you think about creationism being taught in school as a belief.
Could you say that again?
What do you think about creationism being taught in school as a belief?
Sorry, I'm not hit. Oh, what do I think about creationism? Well, I think the debate about creationism is actually a debate about what is science. It's not actually anything else. Science is, of course, an activity which is based upon evidence, thought, and logic. Okay? Now, if you're going to imagine supernatural intervention as an explanation for a phenomenon, it moves into a different realm. It's no longer science. It becomes, let's say, religion. And that's fine. I have no issue with people having a religious belief. I have an issue when religion is pretending to be science, and creationism is not science, because it has to involve a supernatural force, whether it's an intelligent designer, a divine creator, it doesn't matter. What you're saying is, what's responsible for this is something beyond my understanding. Therefore, it ceases to be science. So that's my point.
Okay. Here's my question. What you were talking about a lot before was about the complexity of life and about how all of life really goes into mostly the complex processes that make it up.
Yep.
However, most of the scientific discoveries that have been made so far, and many of the things that are endeavoring to be made, are isolating, say, certain chemicals produced by chemical processes in a chemical system in order to make it have some modern application and modern use in society. So do you therefore think that many of the discoveries will start to shift away from individual chemicals and start to be toward copying entire biological systems for use in commercial use or use in somehow in modern society?
Well, this is a good question. And the point is that science, I used the word, I think this morning, is the art of the soluble. That is, you choose topics that you can solve, that you can deal with. And up until fairly recently, the only really meaningful approaches you could use were ones which did tend to isolate to the smallest components and study those in depth, like the individual chemical reactions in life. And that was fantastically informative. But just now, with the powers of analysis, with the computing power that we now have, we can begin to imagine going beyond that. So it's not that I'm criticizing what's gone before. It's been fantastic what's gone before. And indeed, that's what I've done. But I think now we can look to the future and imagine these more complex ways, and it will be a shift in what we do. So I agree. I think that's the case. Question over here.
I'm just building on what Irina had asked you before, and your answer to the fact that, yes, there could be life forms, but we might not be able to identify them.
Yep.
But do you think there could be common principles or common laws that make sense to us that we could use to identify those kind of new life forms?
So this is an interesting question too, and I think I've got two things to say to that. The first is I think the analysis of these complex systems may generate rules and laws that actually have relevance from aeroplane networks down to living things, and there may be certain common organizational principles that illuminate what life is. The second thing I want to say is something I said in passing. There was a geneticist called Muller who in the 60s argued that the best definition of life was a system that could evolve by natural selection. In other words, he was emphasizing evolution by natural selection as being, if you like, the key diagnostic feature of living things. Mm-hmm. Moved away from the chemistry, moved away from all the description, and just said a system that can evolve by natural selection. And if you're interested in that idea, it's been developed in a book by Cairn Smith called The 7 Clues for Life, I think it's called, published some years ago. And that would be an organizing principle, Muller would have argued, underpins all life. Now, again, I'm not quite sure where I come out on that, but I think it's a very interesting way to think about it.
Thank you.
Good. So, question here, and then we'll go.
You were talking about nonlinear dynamics, chaos theory.
Yeah.
For example, philosophy being applied to theoretical physics. What do you foresee as a limit to sort of these interdisciplinary relationships in different sciences?
Well, I'm a great optimist of both humanity and the human mind, and I think its ability to wrestle with these difficult problems and succeed has been extraordinary. And that's why I said science is truly revolutionary. Remember I said that? It's because it causes these revolutionary changes in our understanding. And I think that we will over the decades, we've got to be humble about it, because life is very complicated, and it's going to be difficult to deal with, but I think we can have a good stab at it. And one of the reasons why the pharmaceutical industry, in my view, is not working as well as some think it should be, we're not getting enough drugs out, is we've underestimated the complexity of living things, including ourselves. And there's a certain humility we need to understand that we need to understand more, but we shouldn't be inactivated by that humility. To follow your point, we should actually take them on, because I think the human brain is really, really effective. Absolutely. Back over here.
Earlier you spoke about how humans' responsibilities to take care of every organism due to our kinship. Yep. However, do you also think that humans should let nature run its own course?
To let it run its own course? Well, I think the question was, just because I'm a bit deaf, I think was that I was saying we have some responsibility as stewards for the biosphere, but you're arguing, should we not let it run its own course? Well, what I think about that is that human intervention in the planet is at such a scale that somehow to let it run its own course gives us, if you like, too much impact on the world. We're having too much influence, is my view. So to let it run its course when we were Stone Age man, I had no problem with, because we weren't having an impact on the biosphere. Now when we see global warming and the destruction of species and so on, it makes me uneasy, and I feel we have to think about our cousins, the rest of life, as well as ourselves. And that has within it climate change and all those issues as well. So I think we shouldn't let it run its own course because we're too dominating at the moment, and it would be too egocentric for the human race. Okay, yeah.
Hi, on the ticket it said you were a Nobel Prize winner. I was wondering what exactly you have researched and what you're presently researching now.
Yeah. Yes, I am. You're right. There's... I'm not the only one. Here's another one here, okay? Anybody else here? I can't think now. I showed you a movie of cells dividing. Do you remember? I worked on the mechanism and the process that controls that and why a cell divides from 1 to 2. I worked it out in yeast, actually, and then showed it works the same in human beings. So that's been the work that the Nobel Prize recognized. I'm now interested not only in more details of that, but also interested, which wouldn't surprise you hearing what I've spoken about, I'm interested in how cells attain certain shapes, how they get spatial order, and I'm also thinking a bit about how systems may work, but I've not had anything very useful to say about that yet. So when I do, I might write something, but not yet. But I'm interested in how cells attain their shape. Spatial order at a distance interests me too.
Thank you.
Okay? Good. One more here, and then I'll go back. Yeah.
With the views and ideas you've talked about today in the fields of biochemistry and chemistry,
yeah,
what would you foresee the average life expectancy to become within the next, say, century?
Right, so the question is how long are we all going to live a century from now? What we've seen is, in New York in 1900, the average age somebody lived to was 50 years of age. Okay? 50 years of age. Now it's about 80. Okay? So over a century, we've seen 30-year increase. It's amazing. I mean, if you think about that. So I see a maybe not such a big increase as that, but a further increase. So over the next century, I suspect people will be living to a fairly healthy way between 90 and 100. I think that may well happen. I do not think that it'll go much beyond that. But there are some people who think it will. So there's some who think that we can go on quite a lot longer than that. But I would see over the next century something like 90, 95 as becoming a sort of not uncommon age. Yeah.
Hello.
Hello.
My name is Waqas Nasser. My question is on intelligence.
Yeah.
From what I think, intelligence is a series of memories which are later put together in different ways by the brain. What do you think about foreign intelligence, say, different forms of intelligence?
Yeah.
And how computers are considered intelligent today?
Well, this is a difficult question, and I don't think I've got a sensible answer, really. I think intelligence is a very difficult thing to fully understand. I think that that computers can do certain things which are very deterministic, though complex, like chess, where there's relatively simple rules, and where you can run through many, many variations on them, so that the computer looks, which plays chess, looks as if it's being intelligent, but it's running through rather simple rules. When we come to the creativity that intelligence in a human being exhibits, I think that's much more difficult to fully understand, and I don't think computers are sort of near that yet. But I'm really not an expert in here. I mean, there are experts in this university who work on neurobiology, neuroscience, and thinking, and it's a very, very interesting problem, but I'm not an expert on that. But I think that computers, my view would be computers are not there yet or even anywhere near it, except in very simple tasks.
How about foreign intelligence, say, like on Mars?
Well, yeah. I mean, imagining, oh, look, behind me, Mars. Did I start that, or did somebody else start that? Oh, you started. This is Mars rover on, looking for something on Mars. Yeah. What does it mean? Intelligence, on, out on Betelgeuse or somewhere. A star. Who knows? Sorry, I have to pass on that one. Yeah.
I was wondering if there's any way that embryonic stem cells could be used or tested without actually having to destroy an embryo.
Yeah. So the question there is, can we use embryonic stem cells without destroying embryos? Well, if we could take adult stem cells, that's cells in the adult body, and reprogram them so they could behave more like embryonic stem cells, that would be a way of circumventing the use of embryos. So, for example, in your skin, you will have cells that can make different cell types that make up skin. Yeah? Now, if we could take such cells and treat them in a way that would reprogram them so that they could make muscle and nerve and so on as well, then that might be a way forward. Now, people are thinking about that, and there's even some experiments out there that are doing it, but we're not there yet. But if that was to happen, then that would be a way of circumventing the use of early embryos. But do remember, the embryos that are being used are very early embryos. They're only, they look like a bag of cells, like that Xenopus embryo. They're usually something like 1,000 cells there. There's no sort of differentiation there. They're normally no more than about 10 days old. They're nothing like a fetus or anything of that sort. So it's very, very early that's being used at the present time.
And with using adult cells like deprogramming them and letting them redifferentiate, is that also a type of cloning?
Well, the cloning part is, you see, one of the interesting things about using stem cells, which is related to cloning, is that you could use cells of your own body, or the nuclei of your own body, to generate these new cell types. And if you do that, then you don't have any problems with rejection of the tissues, the cells that are made, due to the immune system, because it's the same cells as your own body is made up of. So that's one of the big pluses about this. So what people have thought about cloning with respect to this is to take a nucleus from you, let's say, put it into an egg where the nucleus has been destroyed, and making embryonic stem cells. Now, that's one form of cloning. You may have been referring to cloning human individuals.
No.
No. But that is a completely different thing, which sometimes people confuse. But just cloning, in respect I was saying, could generate cells of the same genetic composition and therefore able to produce tissues and organs without any immune rejection.
Thank you.
Thank you. Right, we'll keep going. Back here.
Hi.
If you have to go, just go. That's fine. Don't worry.
My question for you was, who was your favorite 18th or 19th century scientist?
Who was my favorite 18th or 19th century scientist? I've got to go for Charles Darwin. I've got to go for Charles Darwin.
Okay, thank you.
There were some great physicists too, I have to say, but Charles Darwin was a really... read his books and you'll see what I mean. Yeah.
Earlier on you said that the U.S. will move from socialized medicine.
Could you please leave, but do it quietly, all right?
Earlier on you said the U.S. will move from socialized medicine. Are you implying that capitalism or profit-motivated thinking cannot survive or comply with genetics-mediated medicine?
Yeah.
And do you think this will result in a massive corporate-wide struggle to shut down genetics research, justified by the fiduciary duty?
So I don't know if you got that question. The question was, you may remember this morning I said that in my view, the fact that we understand genetic predisposition for disease will undermine private insurance healthcare, ultimately, and that we will have to move to a social-based system. Yeah, now I'm big on that, okay? And I don't think it'll lead to massive corporate breakdown at all. I think you just have to go to Europe and you'll see it's exactly how many systems operate. It just needs a change of thinking about how it's done. Incidentally, the present way of doing it through private insurance in the US is hugely bureaucratic and wasteful of money. 40% of the money, I believe, goes on administration. I think I've got that figure right.
Yeah, it's
That's
like
just
lobbying,
amazing,
and
extraordinary.
which controls what we think about, basically.
Sorry?
Which controls the media and what we think about. So
Well,
we
what do
don't
I
think that.
I think that it needs, there was an attempt, Clinton started to think about it and then gave it up as too difficult. But in the end, the science will force it upon society. Because if you can predict what disease you will get at the age of 37. And unless you keep all of that secret, though it's all out there for you, it's just going to be impossible. So as soon as we get to that point, I think the system will crumble away. And we might as well start preparing for it now. And it's a better socially valuable way to do it, in my view.
Okay. Can I ask one more question?
Yeah, you have another question. I like that one.
Can I have your autograph later?
What was that?
Can I have your autograph later?
Yeah, you can have that later, yeah. Well, one question here, and then we'll go over this side. Yeah. You're a bit short for this, I think.
Hello? Okay.
You're doing fine.
Okay. I was wondering, wow, okay. You know how genes determine how you look like?
In part, interacting with the environment, yeah.
Okay. What do you feel about how people are saying genes are determining our personalities? You know, how a person could be really loud and obnoxious, or, you know, whether they determine they like boys or girls?
Well, this is the point I was trying to make, you see. We are a mixture. We are a consequence of genes and environment. And, you know, it's very obvious the genetic, if you like, effects on physical things like colour of eyes, height, the ability to run fast, things of this sort, you know. But it absolutely also has to be having effects on our personality and our mental abilities at doing different tasks and things of that sort. The point is to sort out how much impact there is in each of these different areas. And always to remember, there's absolutely zero you can do about your genes you inherit, and there's absolutely everything you can do about the environment. So although it may be that there is an impact on genetics, on whether you can do something, you can't influence that, but you can certainly influence environmentally. Let's say you're a runner, okay? You may never be an Olympic runner, but certainly by training you can become quite a decent runner, even if you're not genetically well endowed. So never forget, we can influence the environment a lot. We can't influence our genetic makeup. But understanding this interaction on our personality and the like is really gets to the core of what we think we are, you know? And we have to think about these issues, you know? Think about punishment. We punish people when they do something criminal. If it turns out that their criminality has something to do with genetics, we have to think about that. You know, is punishment the right way to view it? I mean, rather than rehabilitation. It changes the way we think about society, if that's true. Back to the point I said at the beginning, science is truly revolutionary and changes how society works, because we understand how we work better too.
Thank you.
Over here.
Hi. Going back to Irina's question before the first question about possible forms of life that we might not be able to distinguish as of yet, what is your opinion on possibly forms of life that are more evolved than we are, and that's why we can't find them yet? I'm not talking something like a Mars attack or something like that, but more along the lines of more complex life systems that could perhaps have better ways of doing things that could, like, impact our medical system.
Well, you know, I'm not a person who reads science fiction, but the truth is, it's the science fiction writers who have thought more about this in some ways than the scientists, and they, you know, write about life forms like gas clouds and stuff like this. And I mean, it's probably a bit far-fetched, but the only answer I have is that we should let loose our imagination, you know, and imagine what might be out there, because it may be out there. And anyway, that's what science is in part, imagining what might be there. Then, of course, you have to look for the proof, the evidence. And that's what's so powerful about the scientific method. So I could imagine all sorts of things. Whether it's real or not, I don't know. But first of all, imagine, have the creative leap, and then think of ways you can prove it or establish it. And that's science.
Thank you.
Yeah.
Before you said that you believe that within the next century we'll be living to 90 years old.
Yeah, maybe.
And so I want to know, what do you think will be the effect on the environment?
Yeah. Well, I think that just prolonging life without, how can I put it, maintaining the quality of life would be a waste of time. So if we were all just sitting there in wheelchairs for the last 20 years of our lives, I don't think this would be necessarily a high quality way of living. In terms of decaying on the environment, I'm not quite sure of the point you're making, but you may be making that it's going to increase the population of the Earth. And there's no doubt that is a really important issue, the population. And in thinking about climate change and energy usage, into that we've got to factor population control as well. And that we have to think about in terms of how long people live, birth rates and things of that sort. But it's certainly true that if we have the population dominated by the elderly, without people actually keeping things going, then that becomes a social issue that we have to think about.
And also with postponing life, postponing death, do you think that this will have effect on...
Keep going.
Just forgot the question.
Have another go in a minute.
You said that, by the looks of it, that biology is kind of more like physics. In other words, it's being more and more irrational, illogical, and such. So you're predicting...
I didn't quite say irrational and
illogical. I mean like that. Basically, you're saying that it's kind of out of the realm of usual reasoning.
The common sense, outside the world of the common sense.
So you're saying that it'll eventually come... I'm not going to say it's impossible to understand as things like quantum physics, but it'll be a lot harder for regular people to understand. And really, how are you going to cope with that in teaching people and passing on this information?
Well, that problem still is already with us with physics, actually. And you have a struggle a bit with the physics, unless you have the maths to be able to deal with it. We're not there with biology. I just wanted to give an indication of where things might go, and we're not there. But we are there with physics, and there's no doubt about it that reading popular accounts or school-level accounts of quantum mechanics is not desperately satisfying, really, because you don't quite get to it. And Brian Greene, if you read Brian Greene, you read book after book of Brian Greene, and you're still none the wiser at the end of it. Not because Brian isn't a great writer. He's a great writer, but it's just, you can't get to grips with it. So we've got that problem in physics, not yet in biology, though.
Hopefully it won't get to that point.
I'd say. Yeah.
First of all, I'd just like to say thank you for a great talk today.
No, thank you.
And just to move a little back to the genetics question.
Yeah.
Scientists are perfectly willing to recognize that geographic isolation between, say, different ethnic groups has led to their differences in appearance, for example, say, different skin colors or what it might be. Why is that rarely applied to, say, different intelligence quotients?
Yeah. Well, I think that this is a complicated issue, and you will have possibly been reading about Jim Watson's foray into this area.
Yes, I heard about that.
In recent months. I think I've got a couple of things to say about that. The first is that we should not be naive about what we mean by intelligence. Intelligence is a very complicated thing. And you can try and reduce it to a level which is just too simple. So the IQ test, which is where there's been significant study of the genetics and different population and racial differences, measures what it says it does, an IQ test. It measures certain things that you measure there. That isn't necessarily the same thing as intelligence. It measures aspects of certain sorts of intelligence. I was never any good at IQ tests, for example. I mean, it just wasn't my strength. I'm good at doing other things. I'm not stupid. I'm good at doing other things. So I think that there is a naivety in trying to reduce complex traits like intelligence to very sort of simple tests. And I think that's one big issue here, particularly with intelligence. Having said that, I think we do have to consider and debate differences. I mean, we do have to talk about them. And we shouldn't sort of not talk about them just because things are not politically correct and so on. But they always have to be discussed with proper respect and proper understanding of where everybody's coming from and the complexities that are involved. And we don't always see that in some of these debates. And so I think that my main comment is that the level of sophistication of the debate, the level of understanding and sympathy of the debates have got to be very high up there to be able to deal with this. But we should be able to deal with it, because avoiding the issue is not a way forward either. So I think it's a complicated thing, and I don't have an easy way out. But I do think that it's something we ought to be dealing with in a more mature way than perhaps we're dealing with it up until now. Not a good answer, really, but the best I can do.
Thank you. It's great.
Yeah, did you remember? Yeah.
I want to know about longer longevity and with better medical care, what do you think would be the effects on natural selection and evolution?
Well, this question is, if we keep people alive, does this sort of have a negative impact on natural selection? This was a sort of great interest at the beginning of the last century with the eugenics movement, because people were worried about that issue. I don't think that is such an issue. The genetics of natural selection is obviously crucial, but humanity's moved itself into having, if you like, cultural natural selection, which moves much more rapidly and moves itself outside, normal genetic constraints. What I'm meaning is that the advances in society we've seen in the last 100 years have nothing to do with natural selection. I mean, they're built on that for the last 5 million years, but they've come about as a result of cultural and intellectual developments, which are not tied to direct natural selection genetics. So I'm less bothered about that. Also, we mostly reproduce by the age of, 35, 40. So, I mean, it's irrelevant how long you live after that.
But today they're coming out with medicines and techniques to help people reproduce at older ages.
Okay, I'll give you my argument there. If we're going to survive as humanity, we have to have sympathy for humanity. And if we stop caring for the weak, then we are definitely in trouble. And I think any increase in having people who are around who are in some respects not fully healthy is far more compensated by a more healthy view of society, which will help us in the future. Did that make sense?
Yeah.
Yeah.
Thank you.
Good. Probably we should get near maybe 3 more questions. Yep. Here and then 2 over there.
You talk about we are helping the weaks, so the weaks are still reproducing, and do you think that we're still going to evolve? Are we still evolving?
Well, the question is, are we still evolving? And we are. But do remember that the time scale we are looking at, we have a generation time of, say, 30 years. Okay? I mean, 30 years in the sense of from birth to you producing a child, roughly, say, 30 years. And that means in the last 2 centuries, we've had about 6 generations, And that, in terms of evolving our genetics and evolution by natural selection, is an exceedingly short time. It's hardly having any impact. Where we are now is built on sort of 5 million years of evolution from the sort of from an ancestor a bit like the great apes, and that has a much bigger impact. The smaller, the time that we have now, the generations now, are having very little effect on our present sort of genetic makeup. So I think compared with our cultural and societal impact, it's very little, our present genetics. You don't like that? No?
Oh, so you're not sure?
I'm not sure, did you say? I'm a scientist, I can never be completely sure, but I am saying there's very little change in this period of time, and I'm not so worried about that as perhaps some might be. I am saying that. 2 more questions.
I apologize, but my question may be a bit long. Do you think that part of the controversy related to creationism, intelligent design, teaching the controversy, or whatever they're calling it now, is related to the poor PR is related to poor PR on the behalf of the scientific community?
It really interests me why creationism is such an issue in the US, because it's obviously a bigger issue in the US than it is in many other countries, for example, in Western Europe. And I'm interested in why that should be the case. I think that scientists... I'm going to make a general point, which isn't simply to do with creationism, then I'll return to creationism. Scientists like myself or my colleagues who work here have a responsibility to have dialogue with the public. I do think that, because we are an exercise that it relies on society and the community. We are expensive. We cost a lot of money, okay? And the community has got to want to support us. And we have not spent enough time as a scientific community in having dialogue with the public to say what science is and why it's helpful to increase our knowledge and how it can help increase wealth, increase health, increase the quality of life. We've got to have all of those discussions. And in that sense, I don't think scientists have had a good PR. They haven't taken it seriously enough. So I do think that. I think we should be taking it much more seriously. But scientists often think they're wasting their time if they only spend 1% of their time doing such things, whereas they're being short-sighted because 10, 20 years down the line, they could find that, society doesn't want to support science. And that would be catastrophic, I'd say, for society because of what science adds. So I do think PR is an important issue between science and society. If you ask me about the issue over creationism, I don't think it is a PR issue, actually. I think it's to do with the fact that in the US in particular, there is a very strong, in certain areas particularly so, religious movements who have a more literal belief in, for example, the Bible or other religious books. And if they feel that one part of that is undermined, then everything collapses. In a sense, they're tremendously insecure. So if you look at let's talk about Christianity, though it's not the only one relevant here, it's the one I know best about. Many churches have no problem with evolution by natural selection, absolutely zero problem. Others do because they have a literal interpretation of the religious books. And if you undermine part of it, they feel everything falls away. And so that's the reason why creationism, I think, has been such an issue in those more fundamentalist, literal sort of religious beliefs. And so I think it's less to do with PR than to do with religious beliefs that are very literal and very fundamental, which feel threatened if one small part of it is taken away. And it's less to do with PR and more to do with that defensive attitude.
Thank you.
I was interested, I was thinking about what you said about finding other types of life forms. And if they are forms that we couldn't recognize, would they still be formed from the basic unit of the cell as the basic unit of life? Or would there be a different unit?
Well, I think it's a very interesting question, and it is fantasy, you understand, because we can only imagine. And as I said, science starts with imagining, but then needs proof and testing and so on. I really recommend this book, 7 Steps or 7 Clues to the Life by Cavalier Smith. He imagines a life form built on clay particles, okay? Because he imagines clay particles of a certain structure that can evolve by natural selection, okay? It's to do with the fact that you have different sort of mineral deposits and they can break away more easily, therefore reproduce, and sometimes they're stickier and can grow. You can imagine the sort of thing. It's a very interesting sort of book. Now, what that would mean is that you could imagine a life form built on clay particles, if that was true, okay? And that is one example. If you then read the science fiction writers, like Fred Hoyle and so on, they've imagined life forms of a completely different sort. So that's the other place you can look for it. And the only point I wanted to make is that we, when we look for life, imagine it's built the same way. Built on carbon, built on amino acids, built on nucleotides. And just maybe that isn't true, and we should always keep an open mind. So that's the main point I'd like you to go away. But read Cavalier Smith. No, not Cavalier Smith, Cairn Smith. Cairn Smith, sorry, Cairn Smith.
Thank you.
Okay, thank you very much. Thank you.